dipole moment at the end double bond N changes when you change a wavelength of light. You can go from a hydrophobic trans planar form surfactant to a more hydrophilic, less hydrophobic cis bed form of the surfactant. And when we change the hydrophobicity, we change its liking for the protein, how much it likes to bind to the protein, causing different amounts of change in the protein depending on which form the surfactant is present. The beauty of this is it's completely reversible simply by the switch of the light. The technique used is slowing on neutron scattering. The reason we can see things is because they scatter light, so when we have really small things, such as a protein, on the nanoscale, we need to use something of a smaller wavelength in order to get efficient scattering to detect this and then determine the structure. So we shine an instant beam of neutrons at a sample, and then we have a 2D neutron detector. And from this, we can use a novel technique called shape reconstruction. And this is basically, it treats the protein as a collection of N spherical scattering centers, where N is approximately an amino acid. Two most commonly programs used for this are GA Struct and GASPOR. Basically what these programs do is they take scattering centers and they start in a random arrangement, and they calculate the scattering curve. So if your data you collect, the blue points, the scattering from this model here would be this red line. So what the program does is it rearranges each sphere until it gets an accurate fit of the data, and that gives you the shape of your protein in solution. So the specific protein I'm working with is alpha-chymotrypsin, which has been shown to form amyloid fibrils under certain conditions. And in this case, we looked at the inactive form and the active form. The inactive form is at pH 3, and the protein is inactive, enzyme is active at pH 7. We look at both the pure case and then 3 different concentrations of azotab under both visible and UV light. Once the SAMC is obtained, there's several different ways to analyze it. The first step is usually what's called a DNA analysis. So when you look in the DNA region, you can determine 2 things. You can determine the radius gyration, which gives you an idea of the size of your protein solution. And then you can also determine an I0, which when you plot L/I versus Q squared from the y-intercept, you get the I0, which I'll discuss in a moment, to be related to molecular weight. So if we look at a graph of I0 versus molecular weight for several proteins that we have done flying electron scattering on, we can see that for alpha-chymotrypsin, it doesn't lay on the monomer line. So when we calculate our I0, when we look at molecular weight, we get about a 70% dimer. Since small neutron scattering is an additive technique, if you have more than one species in solution, the intensity of the scattering of each species added together will give you your total intensity. So once the I0 is determined, you calculate molecular weight. Now, as I said, since SANS is additive, it's the weight average for molecular weight. As long as you know the molecular weight of each species, you can then calculate the fraction of each species in solution and analyze these 2 species separately. So once we have our raw data, and then we now can use the protein data bank file of alpha-chymotrypsin from the known crystal structure and calculate its scattering using bioequation. We can subtract that from the raw data and get what we term our oligomer-only data. In the case at pH 3, it's a dimer. We know this from the literature. We know this from our I0 calculation. Once this is done, a pair distance distribution function is calculated. This is basically, again, to give us an idea of 1, the maximum dimension of our protein, and the radius. So these are the pair distance distribution functions, and P of R is the probability of finding 2 scattering centers at a distance R apart. This here will give you the most common dimension. In the case of a sphere, that would be your radius. And this will give your longest dimension, which is your protein. And that's just an example of a protein alpha-chymotrypsin dimer. For the pH 7, from the literature, we know there's a monomer hex from equilibrium. And we can also see that from our small neutron scattering data. When we subtract off the monomer curve, we get this characteristic oligomer peak. And so the green curve is basically just the scattering from a hexamer. And again, when we do our pair distribution function, we can see that it's definitely different from the dimer based on the maximum dimension we get and also the most common dimension. Once this is done, we use shape reconstruction to basically solve the inverse scattering problem. Instead of calculating the scattering of a known structure, we have scattering and we're trying to determine what the structure is. So for pH 3, we have monomer dimer. So this is just the oligomer only data, because we all know what the structure of the monomer is. So when we compare it to the protein-amyloid file of alpha-chymotrypsin, it's not exactly the same. However, when we compare it to the chymotrypsinogen, which is an inactive form of alpha-chymotrypsin, and we would expect that to be more like our data at pH 3, since alpha-chymotrypsin is inactive at pH 3. We see that we have very good representation of what our protein looks like, and we can compare that to our ribbon diagram. And in the case of alpha-chymotrypsin, the top protein in the file, the proteins associate in what they call face-to-face association. In the case of chymotrypsinogen, you have somewhat of a back-to-side association, which causes a twist in the molecule, which you can see from this video here. They're slightly offset and twisted. When we look at the pH 7 data, the hexamer, we see pretty large structure that's kind of hard to tell what it is in 2 dimensions. But if we use the same twisted idea, we can propose a ribbon diagram of what this ribbon diagram would look like. And we can actually see the 6 individual subunits. And again, it's a little better in 3 dimensions. And we see that there's kind of a twisted corkscrew-like structure. Up here on the right, the structure shown in red, basically there's not one individual solution. So what the program does is it does this 10 times, and then it calculates the probability that there is a scattering center located where these red dots are. So this is the most probable position of the scattering centers, which agrees well with our structure, showing that we have a good fit and good representation of what our structure looks like in solution. We also show the worst fit, just to show that's basically the worst chi-squared is close to the best. So that's just to show, again, the consistency in our data. And we can just the different views, the front and the side view. So once that's all for the pure data, once we add in alpha-chymotrypsin, under visible light, we get hexamers similar to the pure pH 7. Under UV light, we get what appears to be 2 hexamers associating in a slightly offset manner and coiling around each other to form the decamer. And we can see again this corkscrew-like structure and the twist in the molecule from these 2 videos. Now I keep saying hexamer, I keep saying dodecamer, and I keep saying corkscrew structure. Well, big deal, who cares? Well, the significance is related to these amyloid fibrils. As I said, amyloid fibrils grow in units of 6. They start with a slightly unfolded monomer unit of protein and associate 6 at a time until they finally grow into these large amyloid fibrils. So if we compare the size of our pre-amyloid oligomers, they're much shorter than a fibril. However, they happen to agree very well with the size of these pre-amyloid oligomer hexamers. They're the same length scale and the same cross-sectional radius. So once we did all the small angular neutron scattering data, we want to know, well, do these actually grow into amyloids? So several months later, we did some fluorescence microscopy. We did 2 classic amyloid tests. We stained with Congo red, we just looked at Congo red fluorescence, and then we also looked at it under cross-polarized light. For the fluorescence, in the presence of amyloid fibrils, Congo red fluorescence, Congo red can be compared to our azotab molecule. They both have the azobenzene rings. And so this gives us an idea. People know that the Congo red likes to stack in the beta sheet, the cross-beta structure of amyloid fibrils. So this gives an idea of how our surfactant might be interacting with the pre-amyloid oligomers and amyloid fibrils. For the cross-polarized light, if amyloids are present, you will get what's known as apple green birefringence, or basically a green glowing. And we can see this both at pH 3 and pH 7. We have positive tests for amyloid fibrils. So the big deal is, well, we can form fibrils. Many people have formed fibrils. But it appears that we can reversibly unform fibrils. So with small angle X-ray scattering data, very similar to neutron scattering data, just using X-rays, we just take one of our samples in the UV form of the surfactant, and we then expose it to visible light after 1 hour and then after 2 hours. This is of the original spectrum under visible light. We can see that as we step, we expose longer and longer visible light, we step back down to the original state. Now, since beam time is very limited at the X-ray beam, we didn't have time to actually go all the way back. So to see if it was completely reversible, we used bromopheno blue, which is a pH indicator. It gives us an indication of whether we are associating or not. When we see under visible light, we go to UV light, we see a decrease in the absorbance and also a shift, giving us an indication that we are actually associating. And then what we did is a time-based scan to see the absorbance at 700. So basically, this increase here is characteristic of scattering, meaning that we have something large in solution, large enough to scatter light, such as amyloid fibrils. We can form and unform the fibrils simply by controlling our light. So in conclusion, we have monomer oligomer equilibriums, which depend on light. Under visible light, we have a monomer hexamer equilibrium. Under UV light, we have a monomer to decamer equilibrium. These oligomers are corkscrew-like in nature. Two hexamers associate in a slightly offset manner into decamers. These decamers are pre-amyloid oligomers, and they can be broken up back into hexamers with visible light. And I'd like to acknowledge our collaborators, funding, my lab mates, and of course my advisor. And any questions?
Thanks, Andrea. Good job. Any questions? Questions? Go ahead.
Do you have some idea about what the mechanism is in terms of just the equilibrium between the monomer and your ultima when you use light as a way to shift the equilibrium?
It's hard to hear you.
What do you think is the mechanism that allows the monomer to kind of go back and forth between the hexamer and the back of the monomer?
Well, it has to do with the hydrophobicity of the surfactant interacting with the protein. So alpha-chymotrypsin has an active site. It likes to associate by the active site, but when you introduce the azotab, the hydrophobic azobenzene rings like to jump into the active site, so it causes the protein to associate in a different way. So when we change our surfactant, we change the way it interacts with the proteins, and then the way the proteins interact with one another.
From what I gather, you basically characterize the precursor to these fibers.
Yes.
And light can affect the equilibrium. That's one way it can. What's next? How would you exploit it?
How would I what?
How would you exploit this knowledge?
Well, the idea, back in World War I, they actually used to use azo drugs before penicillin was widely used. And so we know that azotab itself isn't toxic to the body. So the idea is if we can break up these pre-amyloid oligomers, the next step is to try and break up fibrils themselves. And if it can, then, down the road, eventually the idea would be to introduce this into the body, and then with fiber optics, which that's a technology we already have, you put a fiber optic and specifically target certain areas so you could break up these amyloid fibrils.
Okay, thanks.
Go ahead.
Where did you actually, as you said, decided to use the neutron beam to try and use, where did you get the neutrons?
We went to the National Institute of Standards and Technology in Gaithersburg, Maryland. They have a neutron source there. Forgot to acknowledge them.
Is this something you could use to look at something like an enzymatic reaction between two proteins and macromolecules?
If you have enzyme and a substrate, what you can do is you can contrast out one or the other as long as their scattering is different enough, and you could look at each one individually by doing either contrasting out or, as I said, you can subtract off the scattering data and analyze them separately.
Are you using a single wavelength light source to transition between the transistors?
Yeah, we do have filters that specifically, we actually use 350 and 434.
What's the differential between the two?
350 and 434.
You said you just use general visible and UV. I was just wondering
we
how
actually
far
use
apart those
350 and 434. Yeah.
Any more questions? Then let's thank Andrea one more time for a great job.